do the Biology 20 Nutrient Identification Lab. Before we get to the lab, we're going to fill in the information on page 20 using the table that we filled in yesterday on chemical tests. First thing we do when we get to the lab is decide which 2 food samples we are going to test and make sure that we write them down. So I'm going to do energy drink and potato. When you get to the lab, fill your beaker half full with water and place it on the hot plate. Turn the hot plate up to number 10 to heat the water before you need it for your Benedict's reagent test. So our first test tube, we're going to put 10 drops of Benedict's reagent. And we'll do the same for the other test tubes. I've already put it in the other one, so we'll continue with our next ingredient into the first test tube. We will also put 20 drops of water. Once we have all of our test tubes ready and labeled, we're going to put them into the beaker on a hot water bath for 2 to 3 minutes. I've done this earlier, so we'll check the results. Our first one, our water, stayed blue. Our second one, glucose, turned kind of a red color. Our unknown was orange. Food 1 was a mustard color, and food 2 turned yellow. Following the information in our booklets, we're going to put 2 drops of water into 2 of the wells on the spot plate, again following the map in your workbook. Next, we'll put 2 drops of unknown. And then food 1, 2 drops. Food 2. Starch only goes into this well, so we have 2 drops of starch. And then we'll put 2 drops of our albumin, which is 100% protein, into this one. Okay, on this side of the plate, we're going to put 2 drops of iodine into each one of the wells. We're going to place 2 drops of Buiret into each of the wells. Onto our first piece of brown paper, we're going to put a drop of water and some lipid. We can definitely see that the lipid soaks into the paper, makes it translucent. The water does not soak in, so it remains opaque. To the next one, we will add our unknown and compare the results of the unknown with our negative and positive tests on the first one. Then we have our food 1 and our food 2.